Assalamu alaikum wa rahmatullahi wa barakatuh. Peace be upon you all. In this video we are going to see how to use the QLab software to calculate the left ventricular ejection fraction and volumes using the 3DQ Advanced software in the QLab 9. First of all, we have to browse from here and choose the volume that we need to analyze. In a previous video we have seen how to acquire the left ventricular full volume in the previous video that was named how to perform 3D TTE step-by-step approach. Now we're going to see how to use this full volume for analysis. First of all, we have to click this 3DQ Advanced. Once you click it, the screen will be subdivided into 4 boxes like that, like the MPR mode as we have seen before. Each box has a certain color. The green box represented by the green button here, the red button is for the red box, and the blue for the blue, which is usually at the short axis level, and the 3D button for the 3D box, which will show you the planes at which you are cutting your full volume. The steps of the software is displayed here. First step is known as end-diastolic reference points. So the software will give you some points to put at the end-diastolic frame. That means we have to choose the end-diastolic frame first. Because this acquisition is usually guided by the ECG, usually the first frame or the last frame will be the end-diastolic volume empirically. Because this is an acquisition that was guided by the ECG gated multiple beat acquisition. So in order to do that, we have to first put our lines empirically in the middle of the left ventricle. We need a 4-chamber view in the green box and a 2-chamber view in the red box, like that. This is the first step, and then we will move frame by frame from here. Frame 1, just click 2, and then the frame 3 will come like this to choose the largest one. So for me the largest one was here. This is the frame number 1. If I use this frame to calculate the end-diastolic volume, I have to first align my lines like that to open the apex as much as I can. And this is one of the advantages of this software, or the calculation of volumes and ejection fraction from the 3D, because I am avoiding foreshortening as much as I can. Now I will put my lines like that. If you notice, the line can be moved by 2 ways. If I hold the line from the middle, it will be a hand like that. In that case, it will move only from side to side and no rotation. If I moved my mouse to the periphery of this line, it will be a hand plus a curved arrow. That means I can only rotate the line. And the same applies here. The blue line also from the middle, up and down, but from the periphery I can rotate like this. So I need to put this line in the middle of the left ventricle and open the apex. The same here. Also the green line should be in the middle of the mitral valve and to the apex like this. And then I have to click on the yellow arrow, that one, and put it at the septum like this. Now I'm ready to click the first one, which is the end-diastolic reference points. I will click it, so the system will give me some points to put. S means the septal attachment at the base here near the mitral annulus. Then L, lateral wall, basal. Then A, the anterior wall, which is here. And I, the inferior wall, which is here. This is the reverse from what we are saying if we are just doing 2-chamber view from the transthoracic approach. Usually the anterior wall will be here and the inferior wall will be here, but this is right left inverted inside this software. And then 1 point only, AP, which is the apex. I'm free to put it here or there. It is only 1 point, so I'll just put it like that. The software give me now end-diastolic volume, which is 95.4 ml, and this is the cost for the left ventricular end-diastolic volume. The next step here is the end-systolic reference points. That means again I have to choose the end-systolic frame, which is usually the smallest cavity. So I have to move frame by frame from here to choose the smallest cavity, which is for me frame 6, for example. Now again I need to open the apex as much as I can. If you noticed, while moving the red line, the image in the red box is changing, because the red box is representing the plane of cutting by the red line. Now it is in the middle of the apex, middle of the mitral valve, so I'm okay now. I will click in systolic reference points. The same points will come: septal, lateral, anterior, inferior, and then the apex here or there. It's okay. Now the software give me a cast again for the end systolic volume, and the ejection fraction was calculated here as 59.7%, and the stroke volume also was calculated. If I need to change or edit any trace here, if I'm not satisfied, I can click here, add reference points, and then I can click here or there at the points I need the software to consider, and I tell him update. Now it included these points, and the ejection fraction is changing a little bit because I chose other points to trace. Then if I am done, I will click finish. That means I am satisfied by the tracing. I can also come back again and add reference points, whatever, but if I am happy with the tracing, I tell the software to sequence analyze this volume. That means it will trace the endocardium throughout the cardiac cycle to give me a moving cast throughout the cardiac cycle, like what's happening right now, and they give me a curve for the volumes starting from the end diastolic volume, which is the largest one, and this is the end systolic volume. Here I can just hide the waveforms to give me the buttons, and then I will play. So this is a moving cast for the left ventricle in 3D with a calculated ejection fraction. I have another options here. I have show segments. If I click show segments, each segment will be labeled by color, as you can see. Once I will put my mouse at any segment of those, it will show down the name of this segment, like this one, basal infralateral. If I moved up, mid infralateral, like that, I can rotate the volume. That segment is the basal anterolateral, you can see, and from the base here there is I, inferior, S, septal, anterior, and lateral. I can also use these buttons to display one box only. If I click the 3D zoom one, it will enlarge this one only, and it's a good option also to save. I can export the image or a movie from using this button to save it for display or for a lecture or whatsoever. Click back here again. I have also different kinds of display. I can use them. Can use slice plane view, just to show me the planes with the cast inside, like this. I also can choose the iSlice view, give me multiple short axes images. Some labs are using this mode for quantification during stress echocardiography, like one volume at baseline, one volume at peak exercise, to save time. And by this method they are cutting the left ventricle in many short cuts, many short axis cuts, to see the subtle wall motion abnormalities. But this is still investigational and not recommended too much in clinical practice, like that. And what is more important is the show report page. This report page will give you many valuable data: the global values in the systolic volume, systolic volume, stroke volume, ejection fraction estimated, and we have the regional function like standard deviation of each 16 segments, standard deviation 12 segments, like that, and we have the regional one as a percentage, and we have the parametric display, which is very important and very well in understanding the wall motion abnormalities, like pulse eye display by timing. This is the time that each segment took to reach the peak, and this is the excursion of each segment, like how far the segments went inside the left ventricular cavity towards the center during systole. And if you noticed here, the average values were in green, so if the segment took more time to reach the peak, that means it is delayed or hypokinetic. Usually the basal segments will show something like that, which is accepted. But if you have something like a red patch over here, that means the anterior wall is hypokinetic or akinetic or whatever. And the same here for the excursion. If the segment was not moving inside, that means it will remain red, but the blue means the segment is moving. And this is a very good way to display the data in parametric or in bullseye display. This is hide report page, and this is hide waveforms like that, and this is how to display the left ventricle alone. Assalamu alaikum wa rahmatullahi wa barakatuh. I hope all of you found this video of benefit, especially those who are interested in 3D echocardiography and those who are using the QLab software in their daily practice. I will be more than happy to receive your comments and feedback and questions as well, either under the video on the YouTube channel or in the 3D echocardiography forum on the Facebook. Thank you so much, and see you in next videos inshallah. Assalamu alaikum wa rahmatullahi.